Alright, calm. AQA A-level biology topic 6 completely patterned there. We're gonna go through all of organisms respond to changes in environments here. Make sure all of that's patterned there. Alright, cool, safe. Alright, keywords here. So you need to know about some keywords. First thing is stimulus here. So this is something that can be detected by an organism. It can be both external and internal, yeah. So it can be like an external thing or an internal thing, yeah. Alright, cool. Receptor. Receptor is an organ or specialized cell that can detect the change that is causing the stimulus, yeah. So whatever change caused that stimulus, the receptor is gonna be able to know what that is, yeah. It's gonna be able to detect that stimulus, you get me? Response here. This is as a result of the stimulus detected by the receptor, a response will be caused, yeah. This can cause a change in behavior. So for example, yeah, if you touch fire, obviously the fire is a stimulus, yeah. It made your hand get burnt, yeah. After that, the receptor it picked up the fire, and then after that you responded in it, so you moved your hand away, yeah. Alright, calm. Taxis, yeah. So a taxis is a behavioral response here that involves movement in a specific direction. So, for example, if something happens here around you and it changes your direction, so it changes movement, yeah, that's a taxis, yeah. So, for example, if an organism moves towards a stimulus, this is called positive taxis, yeah. So you'll see in a minute like phototropism, yeah, where the plant grows towards the light. That's called a positive taxis, yeah. That's a form of taxis, yeah. Alright, calm. If it moves away from the stimulus, it's called negative taxis, yeah. Alright, cool. That's self-explanatory, yeah. Alright, cool. An example of positive chemotaxis is a mobile bacteria moving towards an area with a higher concentration of glucose. If there's more glucose in an area, that means that the bacteria is going to be able to survive easier. Do you see what I'm saying? It's going to find the food easier, yeah? So naturally, yeah, it will change its behavior and make it move towards that food so it can survive. You see what I'm saying? Alright, cool, bro. Alright, kinesis, yeah? Kinesis is a behavioral response that involves changes in movement in random directions instead of towards or away from a specific stimulus. So this is when the movement changes. So the rate of movement, so like it speeds up, yeah? So it doesn't necessarily mean that it moves direction, but what it means is that the rate of moving directions and the rate of moving is increased. You see what I'm saying? So for example, you will see in a minute. So like in kinesis, the speed of movement and the frequency of changing directions increases, yeah? Alright, cool. This response helps the organism quickly encounter new conditions that might be more favorable, yeah? For example, if a wood lice here is placed in a dry area, it will move faster and change direction often to increase the chance of finding a damp environment. Yeah. So what it means here is taxis here would be like if you put it in a dry area and it knew that it was dry, so it walked towards the damp area. Yeah. Kinesis here is that it's already moving around, it's already looking around there. But when you put it in a dry area, it increases the rate at moving around. It starts moving around even more and it starts moving around faster, yeah. Which means that it's gonna be able to find a damper surface here in a quicker time. You see what I'm saying? Which means that it's gonna survive more to retain moisture and that yeah, so it doesn't dry out. You get me? So that's the difference between kinesis and taxis, yeah. Taxis is when it moves towards something, like it changes its direction, yeah. Kinesis is when it changes the rate of change in direction, when it changes the speed, yeah, not direction. You see what I'm saying? Cool. Phototropism, yeah. So plants respond to external stimuli to improve their chance of survival. Phototropism is a response to light. Plant shoots are positively phototropic here, meaning that they grow towards light. So positively phototropic here means that it's gonna grow towards light, yeah. If it was growing away from light, that would be negatively phototropic. You get me? The growth is controlled by a plant hormone called auxin, yeah, or IAA, yeah, or indoleacetic acid, yeah. And this is produced in the tips, yeah. So it's always produced in the tip of the shoot, yeah. And what happens here in low concentrations of auxin, yeah, it makes the cell elongate, yeah. So IA influences growth by affecting cell elongation, yeah. When light shines on all sides of the root, IA is evenly distributed and the shoot grows straight, yeah. So here the light is even, it's moving to all sides, yeah. Hence the auxin, yeah, it diffuses evenly across the whole thing, and so everything elongates at the same rate, so it just elongates up, yeah. My bad, yeah. If it's uneven, so if it's on the right hand side, yeah, what happens here is the auxin moves from the tip here and it diffuses into the shaded side of the root. So as you see of the shoot, yeah. So as you see, the light is on this direction, yeah. So therefore, the shade is on the other side. So the auxin, yeah, these little particles, yeah, they're gonna move to the left hand side to the shaded region of the shoot, yeah. And so when they do that, that then stimulates more elongation in the left hand side. So if it's elongating more in the left hand side, it's gonna bend towards the right like that on the right hand side, as you see here. So it's gonna bend towards the light. You see what I'm saying? So that's how it works. Yeah, that's why shoots bend towards the light. If the light is above it, it will go up. If it's away from it, it will go towards the sunlight. It will go towards the sun because of that situation. You understand? However, if light comes from one side, yeah, IA builds up on the shaded sides, on the opposite side, yeah, causing cells to elongate over there more. This uneven growth causes the shoot to bend towards the light. Alright, calm. Phototropism in roots, yeah. So, roots don't photosynthesize, yeah. So obviously they don't require light, yeah. Instead, yeah, they're gonna need to anchor into the ground to find water better, yeah. Now, I said that in low concentrations, yeah, IAA will enhance cell elongation, yeah. It makes the cells elongate. But in high concentrations, it does the opposite, has the opposite effect. IA is going to inhibit cell elongation, yes? And in the roots here, it has a lot of IA. There's a high concentration of IAA. So therefore, in roots here, it's not like shoots. In shoots, it's going to elongate whatever side it's on. In roots, it's going to inhibit elongation on whatever side it's on. So it's going to stop elongation on whatever side it's on. You get me? So hence, if light hits the top of the root, yeah, IA moves away from the light to the shaded side as normal, yeah, as standard, bro. Yeah, light's coming there, it's gonna move down to the bottom side, yeah. However, since IA in roots slows it down, it means that the bottom is gonna grow slower, so the bottom would be growing up if you think about it, yeah. So it's gonna be growing up at a slower rate than the top is growing down. So therefore, it's gonna move down in total, fam. You get me? Because the root is gonna bend away from the light, therefore, yeah, because it's gonna slow it down. Do you see what I'm saying? Normally, yeah, more time in the shoot, yeah, it's gonna build up on the bottom again, yeah, which is standard, bro, yeah. But then it elongates more on the side with more oxygen, yeah. So it's gonna elongate up. But in the roots here, it's gonna do the opposite, yeah. It's gonna not elongate on the side with more oxygen, yeah. So more oxygen is here, so it's not gonna elongate on that side. So therefore, it's gonna move down, yeah, into the ground, which is what you want, yeah. Alright, calm. Gravitropism in shoots, yeah. IA will diffuse on the lower side of the shoot, yeah. So naturally, because of gravity, yeah, IA is going to be brought down to the lower side of the shoot, yeah. Hence, if the plant is vertical, it will elongate and the part will grow up. So because there's more oxygen on the bottom, yeah, in shoots, it's going to have like an enhancing effect. So it's going to elongate more on that side. So therefore, it's going to elongate more there, which means it's going to bend up towards opposite gravity. You get me? Which is natural because obviously, if you think about it, yeah, where's the sun going to be and that kind of thing? It's going to be on the opposite end of gravity. You get me? Alright, cool. Hence, if the plant is vertical, it will elongate and the plant will just grow up directly. And this is known as negative gravitropism, yeah? Alright, cool. Gravitropism in roots, yeah? So IA moves to the lower side of the roots, hence the upper side will be growing faster and elongating, yeah? So it's going to move to the bottom side of the root, same as in the shoot, yeah? However, because it inhibits cell elongation on whatever side it's on in the roots here, it's going to inhibit it on the bottom, yeah? Which means that the top is going to grow faster, yeah? So therefore, it's going to go down into the soil, yeah? Which is what you need, yeah? So this is called positive gravitropism, yeah, because it's moving down into the same direction of gravity, yeah. Alright, calm. Simple reflex arc here. So reflexes are rapid, automatic responses that protect the body from harm, yeah. They happen without conscious thought, helping organisms to respond quickly to danger. Reflexes bypass the brain, yeah, so there's no decision-making delay. The fast reaction increases the chance of survival. Alright, cool. So this is the pathway, yeah. So effectively, you just need to memorize this kind of pathway. So there's a stimulus, yeah. So a harmful change in the environment, more time is harmful, and these kind of reflexes, yeah. Some people think that they've evolved over time and that kind of thing, but yeah, they're basically instilled to you from birth in it. So you just spawn with these kind of reflexes, yeah. So then the receptor is gonna detect the stimulus, yeah. So it's gonna find that stimulus if that's a hot flame or whatever that is, yeah. It's gonna detect that, yeah, and it's gonna move into the sensory neuron, yeah, which is the one that's attached to the receptor, yeah. And that's going to carry the impulse from the receptor to the spinal cord, yeah? Obviously, you got the central nervous system and the peripheral one, yeah? The central one, if you think about it, yeah, it can't just move through all these little neurons across the whole body, yeah? That's going to take too long, yeah? So therefore, you need something like a spinal cord. That's the reason you have a spinal cord, yeah? It's to move the signals fast across the body, yeah? From like the finger all the way to the brain rapidly, yeah? So that's called the central nervous system, yeah? The peripheral one is all these little neurons that are outside, like the sensory, the relay, whatever, yeah? Sensory and motor, yeah. Alright, calm. Relay neuron, yeah. So after it moves from the sensory to the spinal cord, the relay neuron located in the spinal cord passes the impulse from the sensory into the motor neuron, yeah. And then the motor neuron is going to carry that impulse from the spinal cord to the effector, yeah. The effector is normally a muscle or a gland, yeah, that carries out a response, yeah. And then the response is obviously the action, yeah. So if you touch a flame, the thing is automatically going to go through the receptor to the sensory neuron, into the motor neuron, and then to the effector, and you're going to pull your hand away from it. You get me? Alright, calm. So this is a simple reflex arc that you can use here to memorize. So you touch the cactus here, and bang, like that comes through the sensory receptor here as pain, through the sensory neuron to the central nervous system there. That relay neuron, yeah, moves it from the sensory neuron to the motor neuron, yeah, and then it goes through the motor neuron, yeah, into the effector, which is your hand, yeah, that moves away from it. You see that? So pretty simple, bro, yeah. Alright, cool. Pacinian corpuscle, yeah. Pacinian corpuscle is a sensory receptor located deep in the skin, particularly in areas such as fingers and whatever, yeah. At its core, yeah, it contains a single sensory neuron, yeah. So it is in a sense like a sensory neuron, yeah, with tissues called lamellae separated by gel. This structure allows it to direct mechanical pressure and vibrations, yeah. The sensory neuron's membrane contains stretch mediated sodium channels, yeah. So basically, yeah, in simple, what happens is it's like this kind of thing in your finger. And so when you touch something, yeah, that pressure causes these sodium channels to open, which is going to lead to depolarization. I'm going to talk about that more in a minute, yeah. But essentially, all it is this is how you detect pressure on your skin. This is why when you touch someone, yeah, you feel something. It's because of the Pacinian corpuscle, yeah. That's why you feel certain shit, yeah. Alright, cool. When there is no pressure, yeah, these channels, these sodium channels, they remain closed, yeah. However, when pressure is applied, the lamellae are deformed, which in turn deforms the neurons and membrane, yeah. This opens the stretch-mediated sodium channels, allowing sodium ions to enter the neuron rapidly, yeah. The influx of positively charged sodium ions changes the membrane potential, leading to depolarization, which is I'm gonna talk about that in a minute, yeah. But effectively, what it means is that when these sodium ions move inside, it carries charge here, and it's gonna carry an electric signal from the finger, yeah. If the depolarization reaches the threshold, it generates a generator potential here, which then triggers an actual. Yeah, okay, cool. Alright, safe. So, as you see here, layers of Pacinian corpuscle here, there's the axon there for the sensory neuron, yeah? The pressure, there's an excess of sodium ions outside the axon, yeah? So when there's no pressure, it's outside, it's standard, bro, yeah? When there's pressure, these sodium ions move into the thing, and when they move into the axon, yeah, it's gonna create that potential. It's gonna create a charge inside the axon, yeah? It's gonna create a signal, yeah? A domino effect here. Alright, calm. The retina, yeah. The retina is a light sensitive layer at the back of the eye that converts light into electrical signals, yeah. So this is the retina over here, yeah. Alright, cool. It contains photoreceptor cells, rods, and cones, yeah. Rods detect light intensity and are more sensitive in low light, yeah, while cones detect color and function best in bright light. When light hits these cells, it causes a chemical change that generates an electrical impulse, yeah. The impulse is transmitted through optic nerve to the brain, yeah, where you can interpret as vision, yeah. Alright, cool. So rods, yeah. Rod cells are a type of photoreceptor found in the retina that are responsible for black and white vision, yeah? So that's why in low light, you only see in black and white. You can't tell apart colors, yeah? That's because rod cells contain light-sensitive pigment called rhodopsin, yeah? You need to memorize that, yeah? That's why I highlighted it, yeah? Rhodopsin is the pigment inside of rod cells, yeah? So when light hits it, the rhodopsin is going to break down, and that's what's going to enable it to respond, yeah? And so it does this in even small amounts of light. That's why um in the dark here, you can still see shit, but it's black and white, yeah, because these rod cells, yeah, they can work in like even small amounts of light, yeah. However, rod cells provide low visual acuity, meaning that they do not allow for sharp or detailed vision, yeah. That's why at the nighttime, yeah, you can't really see shit properly, yeah, because you got that low visual acuity. Only the rod cells are working, only the rod cells are responding. You get what I'm saying? This is because bare rod cells synapse with same bipolar neuron, yeah. So the signals from multiple rods are combined. So what that means basically, yeah, is the same neuron, yeah, that's gonna take it, that same sensory neuron, yeah, is connected to like hundreds of rod cells, yeah. So therefore, all those rod cells together, it's not like a nice one-to-one ratio. You get me? It's not like a nice one-on-one connection between the rod cell and the neuron, yeah. It means that one neuron has to focus on all the rod cells, yeah. So therefore, that's why it has a low visual acuity. So it's not as specific. You get what I'm saying? It's like having it's like having 1 pixel, 2 pixels instead of like 1080p. You get me? As a result, yeah, individual stimuli are not distinguished, yeah. So therefore, can't tell where the light is coming from. It just sees it as light, yeah. Cones, yeah. Cone cells are the other main type of photoreceptor in the retina, and they're responsible for color vision, yeah. Cone cells contain a pigment called iodopsin, yeah, and function best in bright light. They are not sensitive to low light levels, yeah. That's why in low light you can't see color because it doesn't break down the iodopsin, yeah. Alright, cool. There are three types of cone cells, each sensitive to one of the three primary colors here. Light red, uh, colors of light, yeah, red, green, and blue, yeah. Cone cells provide a high visual acuity because each cone cell connects to one single bipolar cell, yeah, or bipolar neuron, yeah. So in this case, it's like 1080p, yeah. Each pixel, each like sensory neuron, yeah, is connected to one cone cell each. You get me? So therefore, you're getting the full maximum effect of that. Alright, cool. Distribution of photoreceptors, yeah. So rods are highly concentrated outside the fovea, yeah. So you need to know that for A level, yeah. So just write that down, make sure you know that, yeah. Rods are highly concentrated outside the fovea, and especially obviously we talked about that, yeah. And cone cells are concentrated most densely inside the fovea, which is the central part of the retina, yeah. Now, what I mean by this is on a graph, as you can see, there's the fovea, yeah. The blue thing is the cones because there's bare cones inside the fovea, yeah. But as you see, it dips down in terms of rod concentration, yeah. So there's not that many. There's literally no rods over here, yeah, inside the center of the fovea, yeah. So you get whereas outside the fovea, you get bare rod cells, yeah. And the blind spot, yeah, is where these two kind of overlap. So the blind spot, you got no cones and you got no rods, yeah. So in your blind spot, you can't actually see shit, but your brain kind of fills it in anyway because it knows what's around you, so it can kind of hypothetically fill it in. So therefore, you see it. But there's tricks that you can do and that sort of shit to like, you get me. Alright, control the heart here. So these are the parts here. So you need to know about the sinoatrial node here. So this is the right in the right atrium is known as the pacemaker. So this is what sets the pace for your heart to move and that kind of thing, yeah. The atrioventricular node, the AVN, is in the right atrium as well, but close to the border with the ventricular, the ventricle, yeah. So as you see, there's the AVN, yeah. The AV node here is next to the ventricle, yeah. It's next to the little like border between them, yeah. Whereas the SAN is over here, that's the pacemaker, yeah. Alright, cool. Then you also got the bundle of His here, which runs across the septum. Obviously, that's a septum here. And the bundle of His is located there. It's like it's going to connect into the septum here and run across it down this black thing here. And then these branches here are called Purkinje fibers here, which is going to move across into the ventricle wall here. Alright, cool. The process here. So the heart is described as myogenic here, meaning that it can contract on its own without needing external nerve signals. So it knows to just keep pumping by itself without anybody telling it to pump here. This self-initiated contraction starts in this SAN, and it's known as the heart's pacemaker. The SAN sends out a wave of electrical excitation that causes both the atria to contract at the same time, pushing blood into the ventricles here. So obviously that's atrial systole here. It's going to move from the atrium into and push blood into the ventricles, yeah, down into the ventricles, yeah. It knows to do that, yeah, because SAN is going to send out a wave of electrical excitation that causes it to move down there, yeah, calm. However, the ventricles do not contract immediately, yeah. So, the atria contracted, it filled up the ventricles, yeah. The ventricles should also contract here because that same electrical signal is going to move down into the ventricles now, yeah. But because the layer is non-conductive here, it's going to take time for it to reach the ventricles, you understand? So in that time that it takes here, it ensures that the ventricles can be fully filled up with blood here before they do ventricular systole and they release all the blood into the body, you get me? So this creates a delay, giving time for the atria to fully empty their blood into the ventricles before the ventricles contract. Alright, cool. The wave of excitation is then picked up by the atrioventricular node here located between the atria. After a short delay, the AVN sends the wave down the bundle of His here, which carries into the apex of the heart here. So now that the blood is inside the ventricle here, the AVN is going to send down another wave here, down the bundle of His here, which is this thing over here, this loops inside the septum here. And then that's going to carry the signal down into the Purkinje fiber here. And that Purkinje fiber is therefore going to contract and push here, which makes sure that the ventricle is going to contract from the bottom to the top here. The apex of the heart is just the bottom of the heart here. That's all it means here. So basically what's happening here is, again, little recap here, is SAN is over there. It starts the whole thing. SAN, as you see the orange arrow here, it moves into the atria. Same over here, it moves into the atria. Yeah, it creates, these are waves, by the way, electrical waves here. And that fills up the ventricles here. Then the AVN, which is that there, sends that signal to the bundle of His here, which moves it down the Purkinje fiber here, inside the walls of the ventricle. And then the walls of the ventricle then contract here to push out all the blood across into the body here. And that's how it's automated here. Alright, calm. Sympathetic and parasympathetic here. So the SAN, which controls the heart's rhythm, is regulated by two nerves from the medulla oblongata in the brain here. The sympathetic nervous system sends signals through this accelerator nerve, which increases the frequency of impulses to the SAN. So basically, yeah, if you're talking about the heart rate rising, or you're talking about something that needs to make it faster or something like that, that's through the sympathetic nervous system, yeah? So you need to know about that, yeah? That's through the accelerator nerve. Whereas if you're talking about something slowing down, your heart rate becoming more sedated, that kind of thing, yeah, that's through the parasympathetic nervous system, yeah? So that's through shit like the vagus nerve and that kind of thing, yeah? Which slows the frequency of impulses and therefore reduces the heart rate, yeah? Such as during rest. So you can think of this, yeah, like a car in it. You need both the accelerator, the sympathetic system, and you need the brake as well, the parasympathetic system, yeah? So basically, these having those systems separate, yeah, having two separate nerves here means that it's gonna properly control the car, properly control the heart. You get me? Sometimes you're gonna need to speed up, sometimes you're gonna need to slow down. You can do that within seconds if you have it like disconnected. That's why you need both sympathetic and parasympathetic, yeah. Because if they were one thing in total, it wouldn't be specific in it, so it can't move correctly, yeah. Alright, calm. Response to pH, yeah. So when carbon dioxide levels in the blood rise, the pH of the blood drops here. Obviously, that's because the carbon dioxide is going to dissolve into carbonic acid and that, yeah. That's going to make the blood more acidic, that kind of thing. So obviously, the pH is going to decrease because acidic means it's going to be pH 1, 2, 3, you get me? This change is detected by chemoreceptors in the carotid arteries here and aorta and brain here. These receptors send more frequent nerve impulses to the medulla oblongata in the brain, yeah? So that's obviously through the sympathetic here. So when you're writing, you can kind of know if something's going to speed up some shit. So, for example, if you've got CO2, you need to remove the CO2, so the blood needs to move faster, yeah? So you can talk about that as through the sympathetic, you see what I'm saying? So basically, yeah, the CO2 is inside the blood, yeah, because you've been doing bare respiration, you've been running shit like that, yeah? So the pH is going to drop here. That's going to be detected by the chemoreceptor. And these receptors send more frequent nerve impulses to the brain, basically. And then in response here, the brain increases signals through the sympathetic nervous pathway to the SAN. That's the pacemaker that sets the pace of the heart. And this causes the SAN to make it beat faster, which is going to increase the heart rate. And as a result, blood's going to flow more quickly to the lungs here, helping to remove the excess CO2 and return pH levels back to normal, yeah? You understand? So that's why when you run, yeah, your heart rate increases. Yeah, it's because of this. It's because the chemoreceptors inside the blood, yeah, they know that this is happening, yeah. So therefore it sends it to your brain. Your brain then sends it to the pacemaker SAN to hurry up, yeah. And that's gonna make it move faster to remove the CO2, yeah. Alright, calm. Pressure, yeah. So baroreceptors detect changes in blood pressure, yeah. If blood pressure gets too high, these receptors send more frequent impulses to the medulla oblongata, yeah. So again, this is about sedating the body, yeah, slowing it down. So obviously, slowing down is always through the brake, it's through the vagus, it's through the parasympathetic. You see what I'm saying? So the medulla then responds by sending signals along the parasympathetic nervous pathway to the SAN. So the SAN knows if it's through the sympathetic, it needs to speed up. If it's through parasympathetic, that's the brake, it needs to slow down. You get me? This causes the heart to slow down, reducing the heart rate. As a result, blood pressure decreases back to normal levels. Yeah, alright, calm. Alright, cool. The neuron, yeah. So you need to know about the structure of the neuron. You got the dendrites here, which is going to connect to other neurons, and that's where the synapses are. Yeah, at the end of these dendrites here. You got the Schwann cells here, which are just myelinated cells here, and they make up the myelin sheath. You got the axon, which is the long part here, the axon here, and then you got nodes of Ranvier here, which is basically like in between each Schwann cell here, you got a node of Ranvier. I'll talk about the purpose of that in a minute. Alright, cool. Resting potential here. Certain man find this hard here, so just yeah. Alright, cool. So basically, in its resting state, a neuron is said to be polarized here, meaning that there's no difference. Sorry, there is a difference in charge between the inside and outside of the axon, yeah? So inside is more negative always compared to the outside, yeah? So inside the axon over here, the inside of the axon here is always more negative compared to the outside as at rest, yeah? At rest, that's the case, yeah? How much more negative? It's negative 70, yeah? So you need to memorize that, negative 70 millivolts, yeah? This is called the resting potential, and it's essential because it sets up the electrical conditions here needed for a nerve impulse to occur. Without this imbalance here, neurons wouldn't be able to transmit signals quickly and efficiently across the body here. Alright, cool. So that's the resting potential. So you just need it to be negative 70 here, so that when it becomes positive, it's going to move the current across. You see what I'm saying? That's why. Obviously, it can't be zero. Reason why it can't be zero here, because you might ask, yeah, why does it need to be negative then? If it's just the equilibrium, if it's just a baseline for everything else here, why does it need to be negative 70 here? The reason for that is because in order to achieve that, it works based on like ions, yeah, moving in. And for it to be zero, it means you'd have to have no ions inside there, and then just a shit ton of ions moving inside. Obviously, that's not reasonable in it. So therefore, it's going to be this difference is going to be because there's more ions outside compared to inside. That's why it's always negative and not zero, yeah. Alright, cool. Achieving it, yeah. So you need to talk about how the resting potential is maintained or how it's created, yeah. It's going to ask you that in the question, yeah. So what you need to write here is that the resting potential is created and maintained by sodium-potassium pump here, which actively transports 3 sodium ions out of the axon and 2 potassium ions into it, yeah. Sodium is +1 charge here. Potassium is +1 charge here. So if I've got 2 potassiums moving inside, that's +2 inside. If it's 3 sodiums moving outside, that's 3 plus outside, yeah. Therefore, I've got less positive inside compared to outside. I've got 2 positive inside, I've got 3 positive outside here. So therefore, I can say what? That's why it's negative inside. That's why it's negative 70, because I've got less positive charge. You get how it works here. So that's why it works this kind of way, yeah. The reason why it's potassium and sodium is not really like anything specific here. The reason why it's 2 different things is because it can't be the same shit, otherwise it's going to diffuse through. So it has to be 2 different ions here. So naturally, it's just sodium and potassium, yeah. Alright, calm. This requires ATP, obviously, because active transport, yeah. Most sodium ion channels are closed, so sodium cannot diffuse back in here, while potassium channels are mainly open, allowing potassium ions to leak back out. So not only is 3 sodium ions moved out here, those 3 can't even move back in, yeah? Whereas the potassium that's moved in, it can still leak out. So imagine how much charge is leaking out and how much charge is being retained. Not that much charge is being retained inside the axon, yeah? That's why it's negative here. You see that 3 is moving out, 2 is moving in. So naturally, you already got that imbalance here. You already got less positive inside. And then from that 2 that moved in, yeah, 1 could just, you know what I'm saying, decide to cut and just leak out. So that's another one that's also lost. So that's why it becomes so negative inside, yeah? Alright, calm. This unequal movement of positive ions causes the outside of the axon to become more positive than the inside, creating the resting potential, yeah? So that's why that negative 70 is made, yeah? Alright, cool. Action potential, yeah? So An action potential is the rapid electrical change here that travels along a neuron when it's stimulated. Yeah. At rest, a neuron has a negative charge inside the axon, yeah, as we talked about, yeah, resting potential -70, yeah. When a stimulus is strong enough, it can cause sodium ion channels to open, yeah. So basically, yeah, naturally, these sodium ion channels, yeah, they're gonna naturally pump sodium into the cell. Yeah, this whole thing of it pumping outside, that's because of active transport, bro. Naturally, by diffusion, it should be moving inside because there's a shit ton of sodium outside and there's not that much inside, so naturally it needs to move inside in it according to logic, yeah. But because of active transport, it's doing the opposite. But when there's a action potential, it means that these sodium channels open, yeah, and they do what they're normally meant to do, which is pump sodium inside. So therefore, sodium is going to enter inside the axon, yeah, causing the inside to become less negative and eventually positive, yeah, because you've got so much sodium moving in now, that's going to become positive, you get it? If the change reaches a threshold of -55, even more sodium channels open, yeah, so I'll talk about that in a minute, yeah. This leads to a quick reversal of charge across the membrane from negative, so it was -70 before, yeah, and now it's +40, fam, marking the peak of the action potential. So now that +40 is that charge. When you talk about impulses sent across the body, that's what it means by the impulse. It means a +40 millivolts. That's what it's talking about. Alright, calm. So as you see here, a stimulus was caused here. It's at -70 normal, yeah, resting potential, yeah. Stimulus happened, bro. When that stimulus happened, yeah, sodium ions moved in, a small amount of them moved in, yeah. Now, when it reaches a certain threshold of -55, so it's going to become less negative inside until it reaches -55, then a shit ton of sodium ions are going to open, sodium channels are going to open, yeah, and sodium is going to move inside, yeah. And that's going to create depolarization because at the resting state, we call that polarization, yeah. So therefore, the opposite of that is depolarization, yeah. So that's going to cause depolarization action potential, yeah? And I'll talk about in a minute how you need to repolarize it now so it's ready for the next stimulus, yeah? Alright, calm. So depolarization, yeah, begins when a stimulus causes sodium channels to open, yeah? Sodium ions rush into the axon due to electrochemical gradient because there's more sodium outside than inside, so naturally it's going to move inside. The only reason it wasn't doing that before is because of active transport, yeah, as we talked about, yeah? This inflow makes the inside of the axon less negative, yeah? Once the threshold potential of -55 is reached, even more sodium channels open in a positive feedback loop here. So if there's more opening, then more will open. That's called positive feedback loop here. The inside of the axon becomes positively charged, reaching +40 millivolts here. This marks the full depolarization across the whole neuron, yeah? Nice. Now, obviously it needs to reset here so that's ready for the next situation. It needs to get back to -70, yeah, so it's ready for another stimulus here to come. It's already sent that impulse out to the body, yeah. It needs to send another one if something else happens, so it needs to therefore come back to the resting state, yeah. So repolarization, yeah. After the axon reaches +40, the sodium channels close, so it knows that at +40 it needs to close here, and the potassium channels open. This allows potassium ions to move out of the axon, which starts to restore the negative internal charge. So now the sodium ions are not being moved into the axon anymore, and instead potassium ions are now moving out of the axon, yeah, so it's now losing the positive charge, so therefore it's going to become more negative and negative again inside. That's called repolarization, yeah. It brings the potential difference back down towards the original resting level. The movement of potassium out of the axon is down its concentration gradient. It helps reset the membrane's electrical state, yeah. Now it will go so negative, yeah, that it will become past -70, yeah. The reason for that, it's because potassium channels don't close immediately after repolarization. So once it reaches -70, yeah, it still takes time for those so it's leaking potassium, yeah. Once it reaches -70, the potassium channels don't just close automatically. You get me? It takes time for that to close, yeah. As a result, it's leaking more potassium than normal, fam. Which means that this delay causes too many potassium ions to leave the axon, making the inside more negative than usual. This is called hyperpolarization. The potential may drop below the normal resting value of -70, yeah? To restore balance here, the sodium potassium pump becomes active again, and it does that same shit that it was doing at the start, yeah? Moving three sodium ions out and two potassium ions in using ATP, yeah? This is going to re-establish slowly the resting potential, yeah? Readying the neuron for the next impulse, yeah? Alright, cool. So what I'm trying to say, yeah? Imagine, yeah, that's the axon there, yeah? So this is inside the axon, that's outside the axon, yeah? Come. What's happening here is imagine my man is a potassium pump here. Imagine my man is a sodium pump here. Three sodiums here are being moved out. So there's now three sodium as outside in the resting potential. And two sodiums are being moved inside here. Hence, what's happening here, bro, is that I've got more sodium outside. I've got 3 plus outside. I've got 2 plus inside. So therefore, it's negative 70 here. Yeah, you get it? So therefore, it's negative 70. Yeah. Hmm. Now, when the action potential comes here, because there's more sodium outside compared to inside, yeah. When the action potential comes, it's gonna make the sodium channel open. Remember, this is by active transport, yeah. So normally it doesn't do that. Normally it should be moving into the axon, yeah. But now, when the action potential comes here, it's gonna make my man do what it normally does, which is open and let these man move through there. So now Na is gonna move through into here, and if you're adding positive charge to an area that's already negative here, it's gonna make it less negative. So when it reaches negative 55, that's when all the sodium pumps are gonna open. So now all these sodium pumps that are across the whole thing here, they're all gonna open and sodium's gonna flood inside. So now again, all this positive charge here that's flooding inside here. So now it's gonna move to become positive 40 here. That's depolarization, yeah. So now inside is positive 40 here. That's depolarization. It's positive inside now, yeah. That's the end of the action potential, yeah. Now you need to repolarize it. You need to make it negative again, yeah. So to make it negative again, what's gonna happen, bro, is the potassium channels are gonna open now, and the sodium ones are gonna close. So these sodium ones are gonna close here, and the potassium one is gonna open, yeah. So potassium's gonna open. Obviously, there's more potassium inside than outside here, so therefore the potassium's gonna move through the pump here outside, which reduces the positive charge inside, which makes it more negative. You understand? And hence it's gonna become more and more negative here. However, it's gonna become too negative here because the potassium channel takes a while to close. So as it's becoming more and more negative, it reaches minus 55, minus 70, and now it's all the way down to like minus 90. Yeah. And now at this point, too many potassium ions have left. And hence, what needs to happen now is the normal balance has to get restored again, yeah? So that normal resting potential. So again, it goes back to normal, and 2 potassium get pumped inside, 3 Na get pumped outside. You understand? As normal, and it establishes that resting potential again. You understand? That's just the whole process there. Um, Alright, cool. So, for example, yeah, if you put this into like words, yeah, if you step on something, yeah, the pressure receptors in the skin they're gonna be activated, and they're also gonna be sensory neurons at the same time, yeah. That force is gonna open these sodium channels, and sodium rushes in. That's gonna cause the depolarization. Then, if the negative 55 threshold is reached, yeah, the action potential is gonna start, and it's gonna move even more sodium into the cell, yeah. Calm. The signal is gonna travel through the sensory neuron axon, yeah, until it reaches the spinal cord, yeah, the central nervous system, yeah. When that happens, yeah, you got 2 options, yeah. Either option one is a reflex, yeah. So in the spinal cord, the sensory neuron synapses with the motor neuron directly. So through the relay, goes to the motor, and bang, you jump away from the sharp thing that you touched, yeah. You get me? Or it could do the second thing, yeah, which is your conscious, yeah. So the signal travels up the spinal cord to the brain instead of just straight to the motor neuron, yeah. Then it passes through the thalamus, yeah. A thalamus is basically like an area where all the sensories come together, and it can like kind of coordinate shit. And then it's gonna go to the cortex, yeah, where you're gonna feel the heat, yeah, and decide if you should pull away. So and originally I put heat, yeah, because it was fire, whatever, yeah. But let's say it's something sharp, yeah. You're gonna decide, shit, is this sharp enough to pull away, or can I still hack it? You know what I'm saying? So that's therefore gonna lead to that's obviously through the conscious, you know what I'm saying? So that can come over time, like in MMA if you're boxing someone, and you know what I'm saying, your eyes automatically close when they come close to your face, yeah. But over time you can train it to keep it open. So you're making that conscious decision, yeah. Instead of making it a reflex, it's gonna move through there. Now, also, yeah, the reason why, for example, if someone calls your name, you just back it out straight away, yeah. The reason for that is because this shit can over time become like a reflex. You see what I'm saying? So what's gonna happen next time is it's gonna come through the sensory neuron, yeah. So you're gonna hear your name, it's gonna come through the sensory neuron, yeah, and it's gonna travel straight to the thalamus, yeah, and then straight to the amygdala, yeah, which is gonna store all like the emotional and like past experiences and shit. And that amygdala knows straight away that's a dangerous situation, so it goes straight to the motor neuron, and it's gonna back out the shank straight away. You see what I'm saying? So it's interesting how over time, obviously due to environment, this shit can change, and you can obviously start to like it can effectively become a reflex. But the difference here is that these are just shortcuts that your brain is making. It's not a reflex in itself. Reflex, you gotta be born with that. You see what I'm saying? So like the fire thing, that's a reflex because you're born that way. You see what I'm saying? But anything that you learn over time, It's through the conscious, just like sped up. You get what I'm saying? Alright, cool. Refractory period, yeah. After an action potential is generated in a neuron, yeah, there's a short period of time known as the refractory period, yeah, during which the neuron cannot be stimulated to produce another action potential, yeah, at calm. So obviously, yeah, you can't just like have this shit going on all the time, yeah. There needs to be like a break, do you know what I'm saying? So the refractory period is essential for several reasons, as it ensures that the impulses are unidirectional, so they're moving in one way. If you had multiple shit going on at the same time, it means that shit will start overlapping, bro. It also helps to produce discrete, non-overlapping impulses by preventing immediate re-stimulation of the neuron, which will ensure that each action potential is a separate event. Yeah, my bad. Additionally, it limits the frequency of nerve impulses, which prevents the neuron from becoming overstimulated and allows time for restoration of resting potential through the action of sodium-potassium pump. Yeah, alright, cool. So you need to talk about the all or nothing principle. Yeah, I've already mentioned this a little bit. Yeah. So basically, what this is here is it basically states here that the action potential, yeah, cannot be generated until negative 55 is reached, yeah. So obviously it starts at negative 70, it becomes more positive, yeah, as positive sodium ions move into the axon, yeah. Then when it reaches negative 55, that's when all the sodium channels open. That's called the all or nothing principle, bro. If something stops before then, it doesn't send the signal, bro. So if you touch something and it's not powerful enough, yeah, it's not gonna go to negative 55, so it's just gonna stop there, bro. It's either everything or not everything. You get me? That's why it's called all or nothing principle, yeah? If the stimulus is strong enough to reach or exceed this threshold, an action potential is generated and always occurs with the same size and strength. So it's always from negative 55, then it goes up to positive 40. You get me? It always stops there to make sure that everything's consistent, yeah? And that you feel the same amount of shit. No matter how much stronger the stimulus is, yeah? If the threshold is not reached, no action potential is triggered at all, yeah? Which means that a neuron either fires completely or doesn't at all, yeah? Which helps maintain clear and consistent signals in the nervous system, yeah? Alright, cool. Saltatory conduction, yeah? So this form of conduction, basically, the myelin sheath here is like a non-conductor, yeah. So therefore, the signal, the electrical charge, here, it can't move through that because it's not a conductor. But you got these things called nodes of Ranvier, yeah, which is in between the myelin sheath, yeah, which is obviously each one of these things is called a Schwann cell, yeah. And so what happens here is because the nodes of Ranvier are close enough here, and because the electrical impulse can't just move straight through like here where it's moving straight across the axon, yeah, because obviously the myelin doesn't conduct, it just jumps straight from one node of Ranvier to the other node of Ranvier, yeah, which means that the conduction is faster. If it was going to move across the whole thing, yeah, it would need to move across everything, whereas now it can just jump across like that, which is faster. So the myelin sheath acts as an insulator, preventing ion movement across the membrane in the covered areas. At the nodes, however, the membrane is exposed and contains a high density of sodium channels, allowing the action potential to be regenerated. Yeah? So saltatory conduction enables rapid transmission of impulses across myelated neurons, which is essential for quick responses in the body. Yeah? Cool. There's other factors as well that could affect conduction. Yeah. So we talked about the myelin sheath. Also, a larger axon diameter, yeah, means faster conduction because there's going to be less resistance in the flow of ions. Yeah? If you have a larger axon diameter, there's going to be less resistance in total, which means it's going to move straight through quicker. Yeah. Also, obviously, higher temperatures increase the rate of ion diffusion and respiration, which boosts ATP production for the sodium-potassium pump, helping maintain the resting potential for more efficiently. Yeah. Alright, cool. Synapses, yeah. So what I've written here is how synapses work here. If you just memorize this is taken directly from a mark scheme. Yeah? If you just memorize this shit, then you'll get the marks, bro. Yeah. So synapses here is depolarization of the presynaptic membrane. So basically, when it reaches the dendrite inside that I've obviously been talking about shit connecting. Yeah, like I've been saying that sensory connects to the relay, which connects to the motor. But how does that actually happen? Yeah, it's through the synapses at the end of each junction. Yeah. So what's going on here is that the presynaptic membrane, yeah, so whatever neuron it came from, that's already been depolarized, yeah. So that's already got the charge running through it, yeah. Then when it reaches the end, it causes calcium channels to open, and the calcium ions enter the synaptic knob, yeah, which is this end part here, yeah. So it's going to enter here these calcium ions, yeah. Now, there's already this neurotransmitter, yeah, normally acetylcholine, yeah, which is inside little bubbles like that, vesicles, yeah. And what's going to happen here is the calcium ions, they're going to cause these vesicles, yeah, to fuse with the membrane around it, yeah. When it fuses with the membrane, it's going to release a neurotransmitter that's inside of it. And that neurotransmitter, yeah, is going to come out through here where my mouse is, yeah, and it's going to diffuse across the cleft, yeah. You call that gap between the two neurons a cleft, yeah. So it's going to move across the cleft, and that blue thing is a transmitter, yeah. It's going to attach to the other side. Once it attaches to the receptors on the other side, the sodium ions are going to enter into the axon of the other side, and it's going to lead to depolarization again. So it's effectively like a stimulus. You know how you got the stimulus and it causes the sodium ions to move inside and makes it positive and all the shit that comes through that? It's the same shit here. It acts as like a stimulus from one neuron to the other neuron, yeah? So the stimulus for the first neuron was like touching the fire. You haven't got the fire at the end of the second neuron, yeah, to make it move the other neuron. So instead of having a fire, you've got this neurotransmitter that moves across and it causes the sodium channels to open up in the second neuron, yeah, which causes depolarization again, yeah, by making it positive inside, yeah. Alright, calm. Cholinergic synapse, yeah. So a cholinergic synapse is a type of synapse that uses neurotransmitter acetylcholine, yeah, to transmit impulses between two neurons. So in the test, you can either say it neurotransmitter or you can name it acetylcholine. It's not really needed, but yeah. It's common though. When action potential reaches the presynaptic neuron, yeah, it causes vesicles containing acetylcholine to fuse with the membrane and release into the synaptic cleft. Yeah, we talked about that, yeah. Acetylcholine then binds to receptors on the postsynaptic neuron, causing sodium channels to open and initiating a new action potential. The neurotransmitter is then broken down by acetylcholine sterase, yeah. So that's a little bit extra information you can use in the essay, yeah, and reabsorbed for reuse here. The type of synapse between motor neurons and relay or other motor neurons. So this type of synapse here with acetylcholine here is normally between motor neurons and relay or other motor neurons. So it's normally between neurons, yeah, not between the neuron and the muscle, yeah. So like we just talked about in the previous slide, yeah. Now, where it touches the muscle, that's called a neuromuscular junction, yeah. A neuromuscular junction is a special type of cholinergic synapse here that occurs between a motor neuron and a muscle fiber. Acetylcholine is still released into the synaptic cleft here, but it binds to the receptors on the muscle membrane and triggers a response directly here. However, instead of another nerve impulse, this causes muscle contraction. So instead of just moving a nerve impulse from one nerve to another nerve here, it goes directly to the muscle. That's what this junction is here. So it always leads to excitatory response here and has more receptors than a typical synapse. And the postsynaptic cells is a muscle cell. It's not a neuron, yeah? So it's not just a neuron on the other side, it's a muscle cell, yeah? Alright, cool. Inhibition, yeah. So inhibition is when obviously the synapse is not going to work anymore, yeah. So certain drugs can lead to that. So this happens when inhibitory substances, yeah, or neurotransmitters, yeah, such as GABA, are released from the presynaptic neuron and bind to receptors in the postsynaptic neuron, yeah. So these neurotransmitters use chloride ions to enter the neuron and potassium ions to leave. This makes the inside of the postsynaptic neuron more negative. So normally, yeah, normally the transmitter would move across here and make sodium pumps here move the sodium into the cell to make it more positive, and obviously that's going to be um depolarization, right? But some neurotransmitters, they cause chlorine ions to move inside instead of sodium ions, yeah? And chlorine ions are obviously negative. So that's going to make the axon negative instead of positive. It's meant to become positive so it can move the charge, yeah? Depolarization. But instead it's going to make it negative, which is hyperpolarization. Remember when it went below the graph, when it went like below into the negative areas, yeah? As a result, more sodium ions are going to be required to get depolarization due to the all or nothing principle. So since it needs that negative 55 to be reached here, it's going to need more sodium ions to bring it back up from whatever it is now, negative 90, negative 100, yeah? Because the chlorine ions back up to negative 55. You see what I'm saying? So that's why that kind of thing happens here. They can ask you that in a test as well. This is kind of the structure you need. And yeah. Spatial summation, yeah? In spatial summation, multiple presynaptic neurons here release neurotransmitter at the same time onto one postsynaptic neuron, yeah? The combined effect can be strong enough to reach the threshold and cause an action potential. It allows weak signals from different neurons to join and create a response here. So spatial here is when like different neurons, like a lot of different neurons, move small amounts of neurotransmitter across here, which might build up and create action here. Whereas temporal here is when one presynaptic neuron releases neurotransmitter many times in a short period here. If these signals come quickly enough, they build up in the synaptic cleft here. So it's like dripping water into a cup. If it's fast enough, the water is going to overflow, yeah? So from many different sides here, it's going to move into one postsynaptic neuron, yeah? Which means that if there's enough of it, it's going to create a action potential, yeah? Alright, calm. Alright, safe. Skeletal muscles here. So you need to know about a couple keywords here. Like tendons, for example, is non-elastic tissue which connect to the bones here. Ligament is elastic tissue that joins bones together and determines the amount of movement possible at a joint. Joints here are the area where the two bones are attached. So basically, to make them move, you need a joint in between them here so it can move here. And then antagonistic muscle pairs here, these are muscle pairs that pull in opposite directions here. So the muscles that go opposite directions, you get me? Flexors and extensor, yeah? Alright, cool. So this is basically the average myofibril, yeah? So this is what it looks like. So basically in a muscle, yeah, you got myofibrils here, and these myofibrils, they got these units here. So from one Z line here to one Z line, that's called a sarcomere, yeah? And these sarcomeres, you got an M line in the middle, you got a Z line from there to there, and basically, the middle is called a H zone, yeah? This is called a Z line. This is called a Z line. The M line is in the middle. And then you need to know that as well. The area around the Z line is called the I band here. The area between the H zone and the rest of it is called the I band here. And in the middle is called the A band here. So you don't need to know much here, basically. It's just that if they give you like a picture like that, yeah, the Z line is there, Z line is there. The middle is the M line. And that's the H zone in the middle, yeah. That's what you need to know. Alright, calm. Now this sarcomere, yeah, so this is another example, yeah. So as you see here, we've got the myosin filament, yeah. Myosin is a part of the filament. So inside of it, so basically you got a muscle, yeah. Muscles are made of microfibrils, yeah, myofibrils, yeah. Inside the myofibril, you got repeating units of sarcomeres, yeah. And in that sarcomere, you got two main like meaty substances, yeah, two main kind of proteins, yeah, which is the myosin and the actin, yeah. So myosin filaments, actin filaments, yeah. And so they overlap in that kind of sense here. The red is the myosin and blue is actin, yeah. So basically, the M line goes through the middle. Yeah, I talked about that. And then the A band goes around that like that. And then I band and then the Z lines are on the opposite side, yeah. So you need to know about that. Cool. So this is basically like the structure inside of a muscle, yeah. So you got the mitochondria there, you got the myofibrils moving through it like that. And you got this thing around it called the sarcoplasmic reticulum, yeah. Now you need to know that these tubes are wrapped around the myofibril, yeah. And all they do is they're like tanks here that contain calcium ions. They're like tanks of calcium ions, you understand? So when it's needed, calcium ions will move from there into the myofibril, yeah, into the sarcoplasm, you get me? So that's basically the situation, yeah. Alright, calm. This is a little close up here of actin and myosin, yeah. So actin is this whole yellow thing, and myosin is the pink thing, yeah. And so the myosin head comes above the myosin, so it comes out of the myosin, and it basically needs to move the actin filaments. That's why your muscle contracts. Your muscle moves like when you flex here. The reason why your muscle fibers move is because actin is sliding on top of myosin, yeah. And how that happens is basically the myosin will grab onto the actin, and it will move the actin forward like that, like it's rowing something, yeah, like rowing a boat, yeah. Alright, cool. You also got inside the actin, yeah. You also got troponin, yeah, and tropomyosin, yeah. Tropomyosin is like basically like a cover, yeah, so that the myosin it will attach to that. It can't attach to that and move it from there. It needs to attach to the actual actin itself, yeah. So that it doesn't always do this. It only does this when, because you see, if the myosin attaches to the yellow part, yeah, it will just start rowing the boat straight away. Do you know what I'm saying? So there needs to be a protective cover over it, yeah, to stop it from doing that. That's why you got this tropomyosin. That's there when it doesn't need to move anything. So the myosin head is attached to that and it doesn't need to do anything. But when it needs to do something, it will move and it will attach to the yellow part. I'll show you in a minute. So muscle contraction, yeah. By the way, if you don't understand this, I've written as well in a couple slides, you'll see it's got the mark scheme version in it. So you can just memorize that as a mark scheme, yeah. And then you can use that in exams. If you don't understand muscle contraction, you'll still get all the marks here. Alright, cool. So basically, yeah, action potential arrives here. So when action potential, a muscle fiber, it triggers a response in the muscle here. So basically, yeah, what's going on here is that there's a whatever response here, sensory thingy here, stimulus here, and as a result, it's come through all the way to the neuromuscular junction here. And now what's gonna happen is that the muscle needs to react to that. The muscle needs to contract here. So what's gonna happen is the action potential is gonna arrive to the muscle fiber, yeah, calm. Then the calcium ion is gonna release here. Calm. The calcium ions, I told you, they're stored inside the sarcoplasmic reticulum, yeah, those tubes, yeah, and that's gonna move from there into the myofibril, into the sarcoplasm, yeah, around the myofibril, yeah. And then these calcium ions, they're gonna bind to the protein troponin, yeah, causing tropomyosin to move and expose binding sites. So what I'm saying is that in default position, yeah, the myosin head is attached to this here, and it doesn't do shit. You understand? It doesn't do shit, yeah. But when the action potential comes, it releases calcium ions, which cause this to kind of change shape and move away to expose this yellow part underneath it. And then the myosin head is going to attach to the yellow part, to the actin. You get it? As you see in the diagram here on the left, calcium ions bind troponin, causing tropomyosin to move out of actin myosin binding site. So that's going to move away now. And you see how now it's exposed. You see how the yellow part of the actin here is exposed to the myosin here. So the myosin is going to attach to that actin here. Calm. Cross bridge formation, yeah. With ADP attached to the myosin head, the myosin binds to the exposed actin site, yeah, forming a cross bridge, yeah. So what's going on is the thing has moved away, yeah. The tropomyosin, yeah, the protective cover, yeah, has moved away, and now the myosin can freely attach to the actin, yeah. So now it's attached to the actin freely like that, you see, and that's caused a cross bridge, yeah. That's made a cross bridge between them. It's fully attached now, yeah. Calm. Then you got a power stroke. The myosin head pivots here, pulling the actin filament along and creating tension here. This movement is called the power stroke. ADP is then released here. So what's going on now is this myosin head moves forward here, it pivots forward and it slides the actin above it forward. So the actin is going to be moving left as you see here according to the diagram here. And then what's going to happen after that is ATP is going to come and hydrolyze, and that's going to return the myosin back to its original state. So detachment, a new ATP molecule binds to the myosin head, causing it to change shape and detach from the actin, yeah? Then ATP hydrolysis, ATP hydrolase is going to break down ATP into ADP and PI, and that's going to release energy, which allows the myosin head to return back to its original position, yeah? And then the cycle is going to keep repeating as long as there's calcium ions coming through, so as long as there's action potential is here, and there's a stimulus, it's going to do that again, and it's going to move it further along, yeah? You get the whole point here. So you can think of this like an analogy, yeah? But before that, it's just a little recap here, yeah? So myosin head binds to exposed binding site on actin, yeah. So myosin head is there, the actin is that thing there, the chains. So normally the tropomyosin would be holding it in place, but because of the calcium ions, it's moved it away, so it's exposed the actin chain now. So now the myosin head can attach to the actin chain, where it says number 1, where my mouse is here. It attaches to the actin, it moves it forward like that, like rowing here, and then the ATP comes here and breaks down into ADP and PI. And that's going to mean that it can move back to its original state, you get me? So like that, and it resets. That's how it works, yeah. So an analogy here is like tug of war, yeah. So the coach is like your nervous system, yeah. It's going to blow a whistle, yeah. It's going to tell the players that it's time to start, yeah. And that's the action potential. That's a signal that tells the muscle, yeah, that it's time to start the process, yeah. So the rope is surrounded by a locked safety cover called tropomyosin, yeah. So that safety cover, imagine that rope, yeah, that you used to play tug of war, yeah. It's underneath like a little coat, yeah. And that coat has a lock on it. Yeah, so the player has to go collect the key, which is the calcium ions, and it's got to remove that cover. Yeah, it's got to remove that tropomyosin. You understand? And once it removes that tropomyosin, then the workers, yeah, it should say the players. Yeah, I don't know why it says workers. Yeah, the players, which are the myosin, can now latch onto the rope. They can now attach to the actin. Yeah, before the myosin couldn't attach to the actin because that tropomyosin is covering it. You understand? But since the calcium ions came through, since they got that key and unlocked the protective cover, yeah, it can now touch the rope in it. So now they've connected and they're ready to pull, yeah? Now, each worker, yeah, they pull the rope towards themselves, yeah? This is like actual power stroke, yeah? Like muscle shortening, yeah? As they do this, this uses up a bit of energy, ADP that they were holding, yeah? Not ATP, they were holding ADP from previously when the ATP broke down, yeah? After the pull, they can't pull again until they let go. To do that, they're going to need a drink for energy, yeah? They're going to need a bit of ATP, you see what I'm saying? So once they drink it, they let go of the rope. They break down that drink, ATP becomes ADP and PI, and this gives them the energy to reset their arm and reach forward so they can get ready to grab it again and move it. You get me? Alright, cool. So this is what you need to write for the mark scheme if it asks you to explain this kind of process here. You just say that calcium ions diffuse into myofibrils from the sarcoplasmic reticulum. Calcium ions cause movement of tropomyosin on actin. This movement causes exposure of binding sites on the actin. Myosin heads attached to the binding site on actin. And then hydrolysis of ATP or myosin heads causes the myosin heads to bend, pull in actin molecules, and an attachment of a new ATP molecule to each myosin head causes myosin heads to detach from actin sites, yeah? So that's all that you need to say for the mark scheme. That will get you the maximum amount of marks, yeah? Normally it's like 5 marks, so you just need to say 5 of these things, and it will give you the maximum, yeah? Alright, slow twitch fibers, yeah? So slow twitch fibers are specialized for slow, steady contractions and are well suited for endurance activities such as marathon running, yeah? These fibers are highly resistant to fatigue, allowing them to function effectively over long periods, yeah? They are adapted for aerobic respiration, yeah, which means that they rely on oxygen to produce energy, yeah? They contain a large amount of myoglobin, which stores oxygen within the muscle, yeah? They also have a rich supply of blood vessels to deliver oxygen and nutrients, yeah? So these are working for, like, slow, steady, you get me, wins the race over time, do you see what I'm saying? Therefore, they need a rich supply of oxygen, yeah, to help them over that long period of time. That's why they got the myoglobin, yeah, that has the oxygen, yeah, which is going to help them with aerobic respiration. You see what I'm saying? Fast twitch fibers, yeah. So fast twitch fibers are adapted for rapid and powerful contractions during short bursts of intense activity, yeah, such as sprinting, yeah. These fibers are designed to release energy quickly but fatigue over a short period, yeah. They are adapted for anaerobic respiration, meaning that they can produce energy without the need for oxygen, yeah? They have a thick and numerous myosin filaments to generate strong contractions, a high concentration of glycogen for quick energy supply, and a high concentration of enzymes needed for anaerobic respiration. Additionally, they store phosphocreatine, which allows for rapid regeneration of ATP, providing immediate energy for intense activity, yeah? Alright, calm. Alright, cool, yeah? Nice. Homeostasis basics here. So homeostasis is the process that maintains a stable internal environment in the body, keeping factors such as temperature, water balance, and pH all steady. Yeah. Obviously, the reason for that is because it's gonna be needed to make sure that the enzymes are working. Yeah. Because if it doesn't do that, then the enzyme's gonna denature. Yeah. So like if it's too hot inside the body, therefore it's gonna denature. So you need something to make sure that's regulated. Yeah. Alright, cool. Homeostasis is mainly controlled through negative feedback here, where any deviation from the ideal state is detected and reversed to restore balance here. Sensory receptors monitor internal conditions and send signals through the nervous or hormonal system to the effectors. Yeah, alright, cool. For example, yeah, if blood glucose level drops here, hormones trigger the conversion of glycogen into glucose here to restore the normal levels. Yeah, alright, cool. In contrast, positive feedback amplifies changes instead of reversing them and occurs less frequently. So, for example, in childbirth, yeah, the cervical dilation it continues to increase. It doesn't, you get me, until you're born, yeah. It doesn't like increase and then stop. Yeah, the body realizes, oh shit, I need to stop that. Yeah, no, it just keeps going, keeps increasing. So that's an example of a positive feedback loop. Yeah, alright, calm. Normal glucose concentration. Yeah, so the concentration of glucose in the blood changes depending on various factors. Yeah, so the factors can include obviously food intake and energy demands. Yeah. Calm. This is essential to maintain blood glucose levels with the healthy range of 7 to 9.9. Yeah, alright, cool. If glucose levels rise too high, the excess cannot be stored as glycogen or fat is instead excreted in the urine. Yeah, excreted in the urine. Yeah, so what that means is that basically one of the first signs of diabetes is that you got excess glucose inside the urine. Yeah, that's a sign that you got diabetes because it can't be stored, so therefore it's gonna move outside. Yeah, alright, cool. The liver carries out three processes to regulate, yeah. Glycogenesis, yeah, which is making glycogen from glucose removed from the blood, yeah. Glycogenolysis, yeah, which is breaking down stored glycogen into glucose, which can be released into the blood, yeah. Or gluconeogenesis, yeah, which is the synthesis of glucose from other molecules such as amino acids or fatty acids, that kind of thing. Alright, cool. So let's say that you had a nice meal, you know what I'm saying? Half pound of 4.99. When blood glucose levels increase, this change is detected by beta cells located in the aisles of Langerhans here inside the pancreas here. So in the pancreas here, you got these beta cells here, which are in the aisle of Langerhans here, and these basically detect the increase, not the decrease, the increase here. In response, the beta cells secrete insulin here, which also works to inhibit the activity of alpha cells. Alpha cells here is going to work when there's too little glucose here. You'll see in a minute. The insulin is then transported through the bloodstream to target cells such as hepatocytes here in the liver, as well as muscle and fat cells. Once insulin reaches these cells, it binds to the specific receptors on their plasma membranes here. This triggers vesicles inside the cells containing glucose transport proteins to move and fuse with the cell membrane. As a result, the permeability of these cells to glucose increases, allowing more glucose to enter. Yeah? The glucose is then either converted into glycogen or fats for storage, or used immediately in respiration to provide energy. So basically, yeah, you've eaten something, yeah, the blood glucose is gonna rise. Hence, the beta cell is going to see that it's going to release insulin, yeah. Insulin is going to move down to the target cell, yeah. And then the target cell is going to, the insulin there is going to bind to the target cell membrane, which is going to make vesicles inside, fuse to the membrane, which is going to make it more permeable to glucose. So glucose is easier to absorb, yeah, and change it into glycogen for storage reasons. You understand? Alright, cool. If blood glucose is too low, yeah, so when our blood glucose levels drop too low, this is detected by alpha cells in the aisles of Langerhans in the pancreas here. You need to know about that, by the way. That's in the aisle of Langerhans in the pancreas here. In response, these alpha cells secrete the hormone glucagon, yeah, which also acts to inhibit the action of beta cells, preventing them from releasing insulin, yeah. So in this sense, it's gonna obviously inhibit beta cells because if it didn't do that, then they'll be making insulin, which is gonna lower it even more, and it's already low, so it needs to increase it. Glucagon then travels through the bloodstream to the liver, where it stimulates hepatocytes to convert stored glycogen into glucose, yeah, through a process known as glycogenolysis, yeah. This newly produced glucose then diffuses out of the liver cells into the blood, raising blood glucose levels back to normal, yeah. At the same time, in order to conserve glucose, other body cells begin to use alternate energy sources such as fatty acids and amino acids for respiration instead of glucose. Yeah, so these things can be turned into glucose or lead to direct respiration in order to conserve that glucose. Yeah, as the glucose concentration is already low, the body doesn't know when it's gonna get that where it needs. You get me? Secondary messenger adrenaline, yeah. So you don't really need to know too much about this. You just need to know the process. Just memorize all the keywords that are written here. So when the hormone adrenaline is released, yeah, it binds to specific receptors on the cell surface membrane of liver cells. Yeah, the binding causes the receptor to change shape inside of the membrane. This change in shape activates the enzyme called adenocyclase, yeah, which then converts ATP into cyclic AMP, or you can call that CAMP. The CAMP acts as a secondary messenger, carrying the signal inside the cell. So sometimes, yeah, if it's direct or it's fast, yeah, you need something to directly tell the cell straight away to start doing shit, yeah. So that's where the CAMP comes into play, yeah. CAMP is gonna move straight into it as a secondary messenger, yeah, and it's gonna cause another enzyme called protein kinase to get activated. And this protein kinase, yeah, is gonna catalyze the conversion of glycogen into glucose, yeah, which is then released into the body to provide energy. This happens during things like fight or flight responses, yeah, that are initiated by adrenaline, yeah. Alright, calm. Diabetes, yeah. Diabetes is a condition where the body is unable to regulate blood glucose levels effectively, yeah. There are two main types, each with different causes and treatments. Yeah, so type 1, yeah, is known as insulin-dependent diabetes, and it's usually develops in early in life. Yeah, it occurs when the body fails to produce insulin, often because of the immune system mistakenly attacks and destroys beta cells in the pancreas. Yeah, so we talked about beta cells. They obviously detect high blood glucose levels, and they obviously release insulin. In certain manner, the immune system can attack them and destroy them, so therefore they don't exist, so they can't make insulin. Yeah, and as a result, people with type 1 diabetes must inject insulin regularly. Yeah, carefully matching their doses to their diet. So obviously, if they're not doing much physical activity, if they're not really eating much, then you can't give them bare insulin. You see what I'm saying? Because their blood glucose levels are already too low. You see what I'm saying? So it's gonna keep their blood glucose levels under control. Yeah. Alright. Cool. Type 2 diabetes, yeah, on the other hand, is non-insulin dependent and typically develops later in life, yeah. It may be caused by reduced insulin production or by target cells becoming less responsive to insulin due to problems with glycoprotein receptors on their membranes. Lifestyle factors, especially poor diet and obesity, are the major factors, yeah. So this is obviously when you don't really do shit, you're eating all this dog shit, you know what I'm saying? You're sitting there on the couch like your uncle on benefits. And over time, you know what I'm saying, your beta cells become immune to all that sugar that you're eating, yeah, and so they don't make insulin anymore because they're not sensitive anymore. You get, so that's why you get this diabetes. Yeah, control of water potential. Yeah, the body must maintain a stable blood water potential to ensure cells don't shrink or soar due to osmosis. Yeah, this helps maintain homeostasis, supports proper cell function, and keeps blood pressure within a healthy range. The kidneys play a vital role in this by regulating the volume and concentration of water in the blood. Alright, cool. So what I'm saying is that the blood, yeah, is unfiltered, yeah, but it needs to be filtered just in case maybe it needs more nutrients, it needs more water, it needs less water, that kind of thing, yeah. And so it needs to go through this whole system here in the nephron, yeah, in order to regulate that. You get me? So water is gonna move through my man, so the blood's gonna move through this whole system where the red thing is, yeah, and so the blood's gonna move here through the afferent arterial, yeah, inside into there. It's gonna move through the glomerulus, yeah, glomerulus, yeah. I don't know why I'm chatting shit. Yeah, and then it's gonna move through the efferent arteriole, yeah. So it's gonna move through all of that, yeah. And around it, wrapping it, yeah, is the Bowman's capsule, yeah. So this Bowman's capsule is where it collects the water from that. So you'll see in a minute. But effectively, the blood moves through here, and it's gonna move down, and it's just gonna wrap around the nephron, yeah. Now, this nephron splits into different parts here. The Bowman's capsule is gonna collect all that water, and then it's gonna move that water, the filtrate, yeah, through the proximal convoluted tube, yeah, down like that. Then it's gonna move it through the loop of Henle, yeah, where shit's gonna be filtered out and it's gonna decide how much water needs to be reabsorbed and that kind of thing, yeah. And then it's gonna move through the distal convoluted tube, yeah, and the collecting duct, yeah, which is gonna go and it's gonna decide how much exactly, like specific filtration. It's gonna say how much exactly of which thing is needed, yeah. How much of this shit is needed, how much of that shit is needed. It's gonna basically remake the blood pretty much, you understand? So the blood moved through here, all the water got drawn out, yeah. The loop of Henle makes sure that the blood is concentrated. It makes sure that the filtrate, yeah, has enough urea and that kind of thing. That's why when you piss, yeah, it's like, you know what I'm saying, it's concentrated, yeah. You understand the situation. So that's why it's gonna move it through that, yeah. And then when it moves it through here to the collecting duct here, that's when it's gonna specifically put in the nutrients that it needs back into the blood, yeah. Put in the molecules that it needs, the protein that it needs back into there. The proteins aren't gonna move because they're too big, but you get the picture that I'm trying to say, yeah. And also how much water is needed exactly, yeah. Alright, cool. So ultrafiltration, yes, this is in the renal corpuscle, yeah. So blood enters the kidney through the renal artery, yeah, under high pressure from the heart. It's gonna flow into the afferent arteriole, yes, that's what I talked about, that thing at the start, yeah, and then into the glomerulus, yeah, a dense capillary network. High pressure forces water and small soluble substances, yeah, like glucose, ions, and urea out of the blood into the bowel's capsule, yeah. So that's that capsule around it that just collects all the water moving out of the blood, yeah. Alright, cool. Large proteins and blood cells remain in the blood as they're too large to pass through, yeah. The efferent arteriole is narrower than the afferent arteriole, maintaining high pressure to aid filtration, yeah. So effectively, what's going on here is this is the Bowman's capsule wrapped around this whole thing. You got the efferent coming in, you got the efferent coming out, and you got the glomerulus coming in the middle. And so what happens here is that small molecules are going to be forced through from the blood in here, from these capillaries into the Bowman's capsule. So through these, it's going to be filtered, ultrafiltration. So only very small things and water can move through, and it just filters the blood completely of water, yeah. Then selective reabsorption, yeah, in the proximal convoluted tube, yeah. So that's the second part, yeah. If you go back and you look at the thing, yeah, as you see, the proximal convoluted tube is here, so the filtrate has moved through here now, yeah. And now what's going to happen is that all glucose and useful substances are going to be reabsorbed back into the blood. You get sodium ions are actively transported from the epithelial cells into blood capillaries, lowering the sodium concentration inside the cells. Sodium ions from the tubule lumen enter the epithelial cells by facilitating diffusion, carrying glucose with them through core transport. So to move this glucose back into the blood, yeah, it's going to need to be moved by core transport. Yeah, if you look at topic, I think it's topic 3 or something, you'll see core transport there. Yeah, and that's all that's going on here. That's all it's basically explaining is that it's going to be reabsorbed by core transport. Yeah, glucose then diffuses into the blood. Urea and other waste products are not reabsorbed and stay in the filtrate. Yeah, so this is the epithelial cell and this is the capillary, yeah? And so it's going to need to move from that convoluted tube, yeah, back into the capillary, yeah? So glucose through cold transport is going to move back in using sodium ions, yeah? And the water is going to move back in as well into the blood, yeah? And that, what this did, yeah, is it removed things like urea and waste. That's what ultrafiltration did, yeah? Alright, calm, yeah? Calm. So loop of Henle, yeah? So you got the ascending limb and the descending limb, yeah? So the whole point of the loop of Henle, yeah, is try and make like to try and make the urine as concentrated as possible, yeah? So try to remove as much water out of the filtrate as possible, yeah? So as much water out of the nephron as possible, yeah? Because obviously the water that's inside there is going to be urine, yeah? That's going to be taken away, yeah? Alright, cool. So the ascending limb uses ATP, yeah, to actively pump out sodium ions, yeah? Creating a low water potential. So the ascending limb, yeah, is not permeable to water. So water can't move through it, yeah? But ions can. So this ascending limb, yeah, moves these sodium ions outside, yeah? And when these sodium ions move outside, it means the fluid around it, yeah, can therefore become like very salty, yeah. And hence, because it's salty, it means that it's gonna have a low water potential because there's a lot of ions. So therefore, water is gonna move from the descending limb outside. So the ascending limb is impermeable to water, so water cannot follow the ions, yeah. The descending limb is permeable to water, but not permeable to ions. So therefore, the ions can't move into the descending limb. They're just staying out here, but the water can move out since there's low water potential outside compared to inside. It's going to move from the descending limb outside. So that means that more water leaves, and hence the filtrate becomes even more concentrated because it's got barely any water now. You see, and then this water that's outside, it can then just be absorbed back into the blood, back into the capillaries that are around this. Yeah, you get me? The descending limb is permeable to water, so water moves out by osmosis into the low water potential area. Yeah. Water is then absorbed into surrounding blood capillaries. At the bottom of the loop, the hairpin, sodium ions naturally diffuse out as well, further lowering water potential. So as well at the bottom in the hairpin, yeah, it means that as well. Sodium ions can also just naturally diffuse out because down there there's a lower concentration of sodium compared to inside as well. So you don't even need to do active transport, yeah. Alright, calm. Mm. There's still convoluted tubule, yeah, and collecting ducts, yeah. Alright, cool. So basically, yeah. Now it's moved all the way to the end here, near the collecting duct here. And now the blood could be a little bit concentrated. It could not have enough water, yeah. So therefore, what's gonna happen here is water continues to move out of the distal convoluted tube here and collecting duct by osmosis as the medulla becomes increasingly concentrated. The collecting duct runs parallel to the loop of Henle, so it passes through the same water gradient, yeah. So what that means is that Same way how outside the loop of Henle, yeah, is very concentrated. The same shit here as well. Outside of the collecting duct, yeah, it's also very concentrated, yeah. Osmoreceptors in the hypothalamus detect drops in blood water potential. So if the blood water potential drops here, the hypothalamus inside the brain, yeah, it basically detects that using osmoreceptors, yeah. And then it releases ADH here from the pituitary gland here. And then what's that gonna do is ADH is gonna travel in the blood and bind to receptors on collecting duct cells, yeah. So basically, the collecting duct here is normally not that permeable to water, so water doesn't really move through it that much here. But when ADH comes and binds to it, it's gonna activate some enzyme here called the phosphorylase here, and that's gonna cause aquaporin containing vesicles to fuse with the membrane. So that's gonna release aquaporin, yeah. And these aquaporin, when they bind to the collecting duct here, it increases the water permeability of the collecting duct, which means that more water can therefore leave the collecting duct here into the concentrated outside area, and hence water is now moved outside, and hence that water then can be moved into the capillaries, bro, to increase the blood potential of the water. Yeah, you understand? And so urea as well can also move outside, which makes the outside even more concentrated, which means that it lowers the water potential even more, so more water potential can move from the collecting duct outside. So basically, the whole point of that is to make sure that like completely no water is left inside that filtrate, is left inside the tubes here, and most of it return back to the blood. You get me? While also filtrating the urea outside, so that urea can go be, you know what I'm saying, used as urine. Yeah, you understand? So yeah, that's it. It's a bit of a long topic here, but it's minor, fam. Alright.